Hey guys, in this video we're going to talk about some of the basic organelles that you'll find in cells. Because we're talking about membrane-bound organelles, we're really going to be talking about eukaryotic cells, because most of these structures will not be found in prokaryotic cells as they lack most membrane-bound organelles, and they lack a key one, which is the nucleus that you see here in this animal cell. So again, with this video, we're going to talk about a lot of eukaryotic organelles, and so really there's going to be one or two that we're going to highlight that will be present in prokaryotes. The first one we're going to talk about is the nucleus, which really is kind of like the command center of the cell. It holds all the genetic material, and what it's going to do is synthesize messenger RNA. And that messenger RNA is kind of like the blueprint for everything that is made in the cell, specifically talking about making proteins in the cell. And those proteins are what carry out the functions throughout the entire cell. So when you're talking about making proteins, it's kind of like making tools or machines to actually do things inside the cell. Without those tools, or robots you might think of like in a factory, the cell won't function. And so the nucleus really holds all this genetic information and is absolutely vital. Again, the nucleus is a key characteristic of eukaryotes, so we won't find these in prokaryotes. If we look at the nucleus, it actually is surrounded with an envelope that actually has two membranes. And that nuclear envelope is actually dotted with thousands of nuclear pores, which allow material to move into and out of the nucleus. Things that move in are building blocks of messenger RNA, so RNA pieces and other things like that. And things that are going to move out are messenger RNA and bits of these blueprints that we talked about with that. And so these nuclear pores become very important because they allow this nucleus to exchange information. But that nuclear envelope will help protect the nucleus from some things that invade the cell. Part of the nucleus is the nucleolus, which is the small highlighted region here. And the nucleolus is actually a grouping of what we call NORs, or nuclear organizing regions. And what they do is they group together, and they're going to be the site of where we make our ribosomes. And ribosomes are pretty important, we'll talk about later, but they're actually made here in the nucleolus, and they'll be shipped out from the nucleolus to the cytoplasm of the cell. And they do that by moving through the nuclear pores. So the nucleolus is a part of the nucleus, and it creates ribosomes. Again, if we take a look at that nucleolus or the nucleus, the nucleolus here is in gray, and it's where your ribosomes are made. And then around that, we have all this chromatin, and that's the genetic material. Again, all eukaryotic cells will keep their actual genome inside the nucleus. We don't find it outside the cell. If we were looking at a prokaryote, they wouldn't have a nucleus. Therefore, all their genetic material would just be throughout the cell. The next organelle we're going to look at are vacuoles and vesicles. Sometimes you'll actually find vacuoles in single-celled organisms, especially paramecium, where you have a contractual vacuole that will help regulate its amount of water. But for the most part, vacuoles are big storage bins. And so what they'll do is they'll store materials such as water, salts, proteins, and carbohydrates. And again, this is the vacuole in a plant cell. Typically speaking, vacuoles are much larger in plant cells. And so they're going to store a lot of water in there, and they're actually going to be pretty important for your plants to stand up straight. If you ever had a flower that leaned over and was wilted, if you knew anything about growing plants, you knew it needed watered. Well, basically what's happened is that flower's vacuole has become empty. In animal cells, vacuoles are much smaller, but they perform the same function. They store materials such as water, salts, proteins, and carbohydrates until they're needed. Vacuoles are also found in unicellular organisms, like we were talking about this paramecium. And like I said before, it'll use that contractual vacuole with this rhythmic contracting to pump excess water out of the cell, which is actually a really important use. This being an animal cell, we're going to use it as a representation for a single-celled organism protist, like a paramecium, because they're like animal cells. They don't have cell walls. They can swim around. And so its vacuole will be very important to it. If we talk about vesicles, these are really small little spheres of cellular membrane. And what vesicles do is they're going to contain structures or materials that either are traveling from one location to the other or are needed for some function in a certain area. For instance, in nerve cells, we'll actually have signal molecules that will be dropped into a synaptic cleft that are stored in these vesicles. So as soon as the time comes to send that signal, they'll merge with the cell wall and they'll push out their signal molecule. So vesicles are very, very functional. I think of them as kind of like the UPS trucks of the cell. You can pack just about anything into them, and you can ship them to whatever location they're needed. Lysosomes are small organelles that are filled with enzymes, and they function as a cleanup crew. They're going to break down your old organelles that are no longer functioning. They're going to break down carbohydrates and invading viral particles that are inside cells. And these lysosomes, they're pretty interesting because really they're kind of like a vesicle that's just stuffed with all kinds of digestive enzymes. They're pretty nice because they're going to clean up the cell and keep it from overcluttering. Lysosomes play an important role in cells in that they keep it clean. Just like your room, if it doesn't stay clean, it becomes less and less functional the dirtier it gets. Same thing with the cell. Next up, we're going to talk about the cytoskeleton. And the cytoskeleton is a very, very complex system of filaments that make up the cell. And if we look here, what we're really looking at is a false color image. So I can kind of outline one cell for you. They've actually taken the nuclear material, which is your DNA, and they've colored it blue. So you'll see that right here. Kind of hard to see when I'm using blue, but that's the nuclear material right there. That's the nucleus. And what you're going to see here, as I highlight in red, is some of the cytoskeletons. And these are microtubules, microfilaments, and intermediate filaments that are all throughout the cell. And basically what they do is they're going to give the cell its shape, and they also allow for mobility. So what will happen is sometimes cells will take this microtubule, and what they'll do is they'll build it. They'll keep building on and on and on. They'll just add on to it. Well, what that does then is it takes the cell and stretches it out. So you can kind of see how this if we were building microtubules here, they'd be like they're stretching out to reach for something. Amoebas are actually a great example of this because they have this amoeboid movement in this exact fashion. They're going to build their cytoskeleton out until it reaches for something. Again, there are three main parts that make up the cytoskeleton: microtubules, microfilaments, and intermediate filaments. We're not going into detail of what those are in this video, but I just wanted you to know the parts of them. The cytoskeleton is also used sometimes like an intracellular highway to where things are actually going to move along it. So for instance, if we talked about something that was maybe over here in an endoplasmic reticulum that needed to be shipped somewhere in the cell, it'll be moved to a Golgi, and then that Golgi would shoot a vesicle, and that vesicle would move along the cytoskeleton. It's pretty interesting how they tend to cling and move along set pathways. So the cytoskeleton has a lot of functions. This picture itself is pretty amazing because, like I said, what they've done is added color to cells, so they're a false color image, to let us see that cytoskeleton that's in green. Microtubules become pretty important because we have pretty much microtubule organizing regions called centrioles. And these centrioles are typically located near the nucleus, although you're not going to see them visible during normal cell life. You're really going to see them during mitosis, or when the cell starts to copy itself. So what I've got here is a phase contrast microscope image that has your chromosomes right here, and they're working towards getting to metaphase. And what you're going to see is the centrosome is actually a location of where your centriole is at. And so you have, these centrioles that are actually making microtubules that are going to connect to our different chromosomes. And so these microtubules that they'll connect with actually act as kind of a rail system so that you can eventually pull these chromosomes apart. This is absolutely important because without this system, your chromosomes couldn't separate and your cells wouldn't be able to divide. Which means, number one, you would never have been, because you started off as a single cell, and through cell division, the process called mitosis, which we'll cover later, you became the entire organism that you currently are. And so without these microtubules, and then specifically the microtubule organizing regions called centrioles, cell division would not be possible. Ribosomes are almost one of the most important things that we'll talk about because ribosomes are really small particles of ribosomal RNA. Remember that they're made in the nucleolus. But what they're going to do is they're going to synthesize protein. And by synthesize, I mean create. So they're going to take coded instructions, those blueprints from your nucleus called messenger RNA, and they're going to create proteins. This is absolutely imperative because without proteins, there would be no functionality in cells. Ribosomes are one of the few things that we talk about in cell structure that are found in both prokaryotic and eukaryotic cells. Up until now, we really haven't run into much that is found in prokaryotic cells widely, but ribosomes are found in every single cell type. And that's because proteins are absolutely important. Ribosomes are going to have mainly two different locations. They're going to be located in the cytoplasm of the cell, or they're going to be located on the rough endoplasmic reticulum. Most of the time, they're actually going to have mRNA connected in the cytoplasm, and if that mRNA has an instruction on it that says, take me to the rough endoplasmic reticulum, they're going to attach to the ER, endoplasmic reticulum, as a way to put that protein right where it needs to be. Ribosomes are absolutely important. These little bits of ribosomal RNA that make up ribosomes create proteins, and that protein is the functional unit of all cells. As I mentioned before, we have this thing called an endoplasmic reticulum, which basically are these little folded membranous discs right here. And notice the ones that I'm highlighting right now have these little nodules on them. See those little dots? Those dots are attached ribosomes. Anytime you have an attached ribosome, you have a rough endoplasmic reticulum. Out here, we don't have any attached ribosomes, and that is your smooth endoplasmic reticulum. Those are our two main parts. Endoplasmic reticulums are pretty important that they're going to modify cells, and they're where lipid components of the cell membrane are created. And that's pretty important because when we talk about a cell growing, it literally is going to create a new membrane, and it has to do that with the endoplasmic reticulum. If we focus on the smooth endoplasmic reticulum, it's not going to have any amount of proteins attached to it. And again, it takes place in the synthesis or making of membrane lipids, and it also has a key role to do with the detoxification of drugs inside the body. And so this organelle on a cellular level is pretty important to us. The endoplasmic reticulum is absolutely key in generation of your cell membrane and the production of many membrane-bound organelles and that are actually, membrane-bound proteins that are created in the endoplasmic reticulum that will eventually find their way to a membrane, or some proteins are going to get shipped outside of the membrane. So this endoplasmic reticulum is very, very important, and again, it's made up of two things. Rough endoplasmic reticulum, which means it has ribosomes, and then of course smooth endoplasmic reticulum, which means it does not have ribosomes.